development with the CSD Solid Form Suite. Just a bit of housekeeping to start with. The presentation will be about 30 minutes long. We will then have a question session. We will send you a recording of the webinar after this. If you have any questions, could you please post them into the chat box in the and send them to all panelists, and Dr. Wood will actually answer them at the end of the webinar. So I'd like to hand you over to my colleague. I'd like to introduce him. He's a key member of the key research and applications team here at CCDC. His research is focused on using knowledge-based methods and theoretical calculations in the area of crystal design and prediction. He's also heavily involved in the Crystal Form Consortium and the ongoing development of the software tools for the Solid Form Suite. And he's now going to tell you more about those tools. Hi, Peter.
Thanks very much, Anne. Okay, so I'll start by saying a little bit about the reasons behind using co-crystals, just to set the scene. I'm sure you're aware of the benefits of co-crystals. Our previous webinar from Dr. Eleanor Pidcock focused on the area of polymorphism, and specifically pure solid forms of active ingredients. For a lot of active ingredients, though, the pure form may not have the required properties for development. The chemical and physical stability may not be ideal, solubility might be a problem, or cross-processing properties like the crystal shape or the tabletability. So it's important to have the ability to modulate the solid form. Beyond that, it's also important to have some knowledge of the wider solid form landscape, the purposes of patent defence or life cycle management. So, if we're thinking about other solid forms that are available for an active ingredient, we could talk about salts, co-crystals, hydrates, solvates, but we're going to focus today specifically on co-crystals. I'd like to say that a lot of the techniques I'm going to be talking about are equally applicable to salts and hydrates and solvates. In fact, one of the examples later on is about a salt system. But the specific benefits of co-crystals over these other multi-component systems are as shown on the screen. So if the active ingredient is non-ionizable, salts aren't an option. So co-crystals are really one of the best ways to go. There's also a very large range of potential pharmaceutically acceptable co-formers, as opposed to a relatively small range of counter-ions available. There's also scope for a very significant range in properties with co-crystals in comparison to polymorphs. So if you're thinking about polymorphs, a paper in 2005 was doing some research that suggested that you're only likely to see a factor of 2 change in solubility, whereas with co-crystals you can experience a lot wider variation in properties. Co-crystals are also amenable to crystal engineering through synthons and CSD-based analysis. So we'll see later on in what ways you can actually use that. So how do we find or identify a co-crystal of an active ingredient? Well, surely experimental co-crystal screening is easy and simple. There are a lot of papers looking at co-crystallization. A couple of well-known examples are carbamazepine and sulfothiazole, and there's 50 published co-crystals of carbamazepine, and over 100 solvates or co-crystals are reported to have been made for sulfothiazole. Even for a larger active ingredient, more complicated chemistry like AMG 517, some studies published on this recently suggest that there's at least 15 co-crystals of that. So if that's the case, then what's the problem? Well, these very much depend on the compound you're looking at. For another compound called artemisinin, which is another pharmaceutical, initial co-crystal screens on this found no co-crystal forms at all. And even with rigorous liquid-assisted grinding experiments, only 2 out of 25 co-formers actually resulted in a new solid form. So this is less than 3%. Other studies have found that it takes 6 months or more just to find a single co-crystal of suitable quality for X-ray diffraction analysis. So it's by no means always an easy situation, and it can be like finding a needle in a haystack. So the good news is whether the example you're looking at is easy or not, knowledge-based methods are there to help. So there are millions of interactions in the CSD, all these intermolecular interactions from real observed experimental structures. So the principle I'm going to be talking about today is that we can use predictive analytics, use this information to find the best co-former leads for a given compound. And what I'd like to stress is that the goal here is not to replace experimental screening at all. What we want to try and do is to reduce the amount of time and resource spent on screening and focus the experimental work on the most likely compounds to give positive results. So the most widely known and used method for co-crystal design is synthon method, as described by Desiraju back in '95. And the principle with hydrogen bonding synthons is that if there's a very specific interaction or motif that can form between two molecules, and if it's sufficiently strong or robust, then this is a reliable unit that we can use for engineering the crystal. In terms of co-crystals, the principle is that the motif between the target molecule and the co-former, this is a heterosynthon, should be competitive with any motifs that are formed by the target molecule itself. So those would be homosynthons. So it's all about that competition. There are other techniques for designing co-crystals, and we'll take a look at each of these during the presentation. So these are some other design techniques, and essentially each of these principles uses existing structural knowledge for design and prediction purposes, just in different ways. So all of the examples I'm going to be talking about through this presentation are based on published research. So we're going to start looking at synthons and then move on from there. So for examples of the first two design methods, we're going to be using the example compound of piracetam. This is a pharmaceutical compound which is a neurotropic drug. And piracetam has been studied quite highly with respect to co-crystals. There are over 10 co-crystal structures published in literature. And what we're going to be looking at today is the results of a recent study by Virtualhaus, Hilfiger and Blatter. And they've reported some results of an experimental co-crystal screening work using a range of 22 co-formers. And in their case, they investigated co-formers that covered a broad range of functional groups whilst still being suitable for pharmaceutical use. So we'll be able to use that information with positive results and negative results from their screening. Okay, so firstly, if we're going to be talking about the hydrogen bonding synthon method, how do we put this in practice with a specific example? So using piracetam as an example, we can look at the hydrogen bonding functional groups that are actually observed in that structure. So in piracetam, we have primary carboxamide functional group, and we also have this oxopyrrolidine group. And the homosynthons that can occur in piracetam solid forms are these two shown here, which are the primary carboxamide to primary carboxamide interaction, or primary carboxamide to oxopyrrolidine. So we can use the CSD Solid Form Suite motif searches, which are easily available through the interface, to find out very quickly and efficiently what the frequencies of occurrence of these motifs are. And the frequency of occurrence is essentially saying, for all of the structures in which these functional groups are present, how frequently does the actual motif we're looking at form. So you can see that for these two homosynthons, there's a roughly 63% and a 46% frequency of occurrence. And what we're going to be looking at next is whether other heterosynthons are competitive with those two. So when we're looking at the 22 co-formers that Virtualhaus's study used for that screening method, and I don't want you to look at the specific numbers too much, but what we're showing here is essentially results of motif searches on every pair of functional groups from a coformer to the target molecule. And we have 3 sets of data showing the coformers as acceptors or as donors to either of the 2 piracetam molecules. So what we can see from these results is that a certain few of the pairs of functional groups are highlighted in red, and these are the ones where the frequency of occurrence is higher or approximately the same as the pure motifs. So essentially, these are the heterosynthons that are competitive with the piracetam pure forms. So what can we do with these results? Essentially, if we take an overview of all of the coformers that Virtual and co-workers used, we can pick out the ones which, using the Solid Form Suite motif analysis, are shown to have competitive interactions with the pure form, and pick out the ones which are indicated not to have any competitive motifs. So it turns out that their coformer set is actually a very good match to piracetam. So we have quite a number of carboxylic acid functional groups which interact very strongly with the carboxamide group in piracetam. But we can quickly rule out 5 of the compounds which are indicated based on the synthon method to not be ideal for forming coformers. And if you imagine you start with a coformer library of 100 compounds rather than 22 in this case, then you could rule out quite a number very quickly. So that was a quick overview of a method of using the synthon approach with the Solid Form Suite. But hydrogen bonds are not the only factor, and I'm just going to touch on the fact that we can actually use CSD data to analyse more aspects of the molecule than just the hydrogen bonds. So this is based on some work done by Laszlo Fabian a couple of years ago, who studied a large data set of co-crystals in the CSD, 973 structures in total. So Laszlo looked at a wide range of molecular descriptors with regards to predicting which molecules will form co-crystals with a given target. So Laszlo used 130 descriptors and then used a statistical process to work out which were the most predictive. And Laszlo found that there were 4 descriptors based on polarity and molecular shape that were the most effective at predicting which coformers would form a co-crystal with the target molecule. So what we can do is take those 4 descriptors that Laszlo identified, which as shown here, fractional nitrogen oxygen atoms, dipole, and then 2 descriptors based on the shape of the molecules. Calculate those for our target, which is piracetam. And then the principle is we calculate the molecular descriptors for each of the coformer molecules and match using some of Laszlo's rules of thumb for molecular complementarity to identify which of the coformers are the most likely to form a co-crystal. And again, we're using predictive analytics here to narrow down the options to try and reduce the amount of time and resource we spend on co-crystal screening. So if we return to the set of coformers that Virtual and co-workers were using, we can already remove the ones identified to be not good for coformers in terms of synthons. And then we can also identify the ones which have not complementary molecular shapes and polarities to piracetam. So again, we can see that actually the data set we started with is very efficient, and the shapes and sizes and polarities are all quite good, quite well suited to piracetam. But we can still remove some more. So what we've done now is we've filtered the data set down from 22 molecules to the 14 most suitable for forming co-crystals. And I should stress that a lot of this is things that as research and development scientists you probably do naturally, or you do this type of rationale in your head, but the process provides a quick and easy way of doing it and some quantitative numbers behind the analysis. So we've reduced down to the 14 most likely co-crystals, and in actual fact, we can identify that 4 of those co-crystals, or 4 of those co-formers, actually formed co-crystals with piracetam in the experiment by Peterhouse. So essentially what we've done is reduce our odds from 4 out of 22 down to 4 out of 14, which is pretty good for a relatively quick analysis. And if you started with a data set of 100 odd co-formers, this is massively reducing the number of experiments to do. So essentially this is a computational and quantitative analysis that you can use alongside the sort of scientific knowledge and rules of thumb that you might already have. Okay, so I've talked a bit about synthons and molecular descriptors. What I'm going to be talking about next is hydrogen-bond propensities and a slightly different way of analysing your possible co-formers. So, hydrogen-bond propensities are related to the concept of hydrogen-bonding synthons, but essentially this is a technique that we've implemented at the CCDC that's a lot more statistically rigorous, as well as factoring in additional information such as functional group competition, aromaticity and steric hindrance, other factors. So I'm going to quickly review the principles of hydrogen-bond propensity analysis. But in actual fact, Elmer discussed the details of this propensity method at more length in the previous webinar. So if you're particularly interested in the details of how this is done, then I'd like to say that you still have access to the recording of the previous webinar online. So how does the propensity method work? So essentially, we provide the target or prediction molecule, and in our case, when we're looking at co-crystals, this is 2 molecules, your target and your co-former. And then we'll go through a number of steps which are all automated in this tool. So the user can interact but doesn't need to. So the functional groups are defined or taken from a library. The CSD is then searched automatically for relevant structures containing those functional groups. Hydrogen bonds in those structures are then automatically analysed to find out whether each pair of functional groups does or doesn't interact. And then a propensity model is fit against the CSD data. Again, this is all done automatically. So at the end, you obtain a model for your data set. And then hydrogen bond propensities are predicted for each possible motif or pair of functional groups. So at the end, we're going to get a number saying how likely for our system each pair of functional groups are to interact. So then we can then efficiently analyse the landscape or the interactions possible for that pair of molecules. And again, I've shown a screenshot here showing where you have access to this tool within Mercury. So all of this is part of the Solid Form Suite. So the example I'm going to be talking about in terms of hydrogen-bond propensities is the compound lamotrigine, which is an anticonvulsive drug and mood stabilizer. So lamotrigine has also been analysed and studied with respect to co-crystals because of its poor solubility and dissolution rate in pure form. So what we're going to be looking at is the results of a crystal engineering case study by Cheney and co-workers, and they published some of the negative results and positive results again. So we can look at which molecules do and don't form co-crystals. So what I'm looking at here is 2 of the possible co-formers that Cheney and co-workers used. So at the top we have hydroxyanisole and butane hydroxyanisole, and at the bottom we have methylparaben. And if you look to the right hand side, I've shown some depictions of the hydrogen bond propensities. And because this is a statistical method, what we have here is the range, the bounds of the propensity that's been calculated by the model. So each of the arrows represents one of the functional group pairs. So So in green we have the top and second pure interactions, so the lamotrigine interactions. And you can see these have very high propensities. So these are very likely to form a very strong interactions. And then in blue we have the first and second highest heterosynthons, lamotrigine to hydroxyanisole interaction. And you can see for the top case, there's no overlap between the pure interactions and the hetero interactions. So this is indicating that the target to co-former interactions are not competitive with the pure form interaction. At the bottom, with the methyl paraben case, we can see that again the pure form interactions are very strong and they're given very high propensities based on the model. But we can see that in blue that the hetero interactions are overlapping with the pure interactions. So here we can see that there is definite competition between those motifs in this target co-former model. So if we go and look at the experimental results published by Cheney and co-workers, we can see that in fact no co-crystals were observed with the hydroxyanisole compound. These experiments actually produced a methanol sulfate and no co-crystal. Whereas looking at methyl paraben, two different co-crystals were observed. And here we have images of those two packing diagrams from those two co-crystals at the bottom. It's worth mentioning that for those of you that have used the propensity tool before, we're hoping to speed up and improve the process of doing this for co-crystals in the future to make it more efficient as all as part of the CSD process. So that shows you an example of how we can do a more statistical analysis of co-crystals. But again, that was based on no previous knowledge of a crystal structure. What I'm going to be talking about next is how we can actually use the information of existing crystal structures to help predict new ones. So this is design from a known structure. Essentially this analysis is based on a piece of research by Felipe Martins, which was published a couple of years ago, and is looking at structures of the pharmaceutical compound lamivudine, which is an anti-HIV drug. So as I said before, this particular example is looking at a salt structure and using that information to predict new structures. But the principle is equally applicable for co-crystals. So what we know about this structure is that we have a crystalline salt of lamivudine with saccharinate as the counterion. And the question is whether it's possible to find other guests that fit the API framework without perturbing that framework. So can we design a new form based on this known structure? Can we essentially remove the saccharinate counterion and replace it with something else? So the structure that we can see in the lamivudine saccharinate case is that we have this deprotonated saccharinate counterion in the center, and it interacts with the host framework via these three hydrogen bonds. And in the research study by Martins, they proposed that essentially if we take out this saccharinate and find something else that forms hydrogen bonds in the same manner and fills the same space, we should be able to retain the host framework but still produce a new solid form with potentially new properties. So what we're doing is using some method to try and find a jigsaw piece that can fit into this puzzle, retaining those hydrogen bonds. So the process which Martins and co-workers used was basically selecting a new counterion by arguments of geometric similarity of hydrogen bond acceptors and looking at the pKa of the counterions. I'm going to illustrate how we can use the Solid Form Suite to approach this problem in a similar way, but in a more general fashion. So what we can use is the packing feature part of the Solid Form Suite within Mercury. We can then develop a subset of the CSD containing molecules that are of relevance, so in this case salts with singly charged anions. And what we need to find the molecule that will fit into the framework is that we need hydrogen bonding groups that are in the same orientation. So we can use this packing feature search on the cyclonate counterion using those acceptor points, the ones that we've already identified as forming interactions with the host. We don't need to worry about this second acceptor on the sulfonyl group because it doesn't interact with the framework. So these pink spheres indicate the features of the molecule that are used as a search. And we selected another atom from part of the carbon backbone to fill space. And essentially, the points we've selected here are used as a three-dimensional search of the CSD to find related species in terms of the distances and angles of the acceptors. So what the search finds is a whole range of different counterions that we could use that have geometric relationship of acceptors in the same way. And here are just some examples. I can particularly draw your attention to the malleate counterion on the right-hand side. And it turns out this is the compound that Martin's used in his study. So what they found in their case was that there was indeed a structure of the malleate. You could form a salt of lamivudine malleate, and hydrogen bonding of the counterion to the host framework is in fact the same. And then if we look at another orientation of the structure, you can see just how much similarity there is between the two. Essentially we have simply replaced that counterion in the centre and retained the host framework. So a new crystalline form of the API has been generated purely using the knowledge of an existing form. And again, as I said before, this principle is applicable to not just salts, but also co-crystals and solvates. So even if you have a solvate form of an API that you know you're never going to use for development, you can still use that information that's in the structure to find new forms. On a related note, we can actually use design from known structures in a slightly different way. So a similar knowledge-based approach has been published very recently by Springwell and co-workers. And essentially what they did, they were trying to find co-crystals of levetiracetam. And they used the knowledge that piracetam is a very similar chemical compound and also geometrically very similar. And they knew that there were a number of co-crystals of piracetam. So they used the knowledge of those co-formers in the co-crystal screen for levetiracetam. Essentially, instead of doing a full screen, they used just the 10 co-formers that were known to work for piracetam. And they found 4 new co-crystals from just 10 grinding experiments. So you're getting a return of 40% from the screening experiment. So this is another just to highlight again the power of using existing knowledge when you're setting up your design experiments. So what this builds up to, all of these tools that I've been talking about, is an effective hierarchy of co-crystal design tools. At the top we have the synthon and shape-based methods, which can effectively narrow down a large set of co-formers to the ones most likely to produce results for your target compound. Then we have the propensity method, which can provide a much more accurate and sophisticated prediction for a smaller subset of possible target co-former pairs. And then, if you have some experimental structures, either of multi-component forms of the target or of multi-component forms of a related compound, you can use three-dimensional information in these for design purposes as well, in the ways that I've discussed. So, just before finishing, I want to stress at this point just how much the development of the software tools I've been talking about has been thanks to the input of research and development scientists from our 11 Crystal Form Consortium member organisations. The industrial insight we've obtained from the CSC members has been invaluable, and the CSC programme is continuing to go a close link with the development community into the future. So the continuing goal of the CSD is to develop software tools to make high quality decisions with less risk. So I hope through the presentation I've shown you different ways in which the CSD Solid Form Suite can help you apply predictive analytics to find the best co-form leads. This can really help you to focus on the best experiments to perform, not to replace experiments at all, but reduce the time and resource spent on screening. So finally, the software should help you predict and characterize new forms, and hopefully with improved physicochemical properties. So just to sort of round off, I think if you found this webinar interesting, then please get in touch, ask us about the white papers, request a trial of the software if you don't have it, and please get in contact if you want to participate in the Crystal Form Consortium. And these are the email addresses to get in contact. So thank you very much for listening, and I'll pass you back over to Anne.
Okay, thank you very much, Pete. That's really been very useful expose of how to use the tools in this area. My colleague Pete